Thank you very much for, first of all, for the invitation, and it's really nice. And I'm really sorry we can't be really face to face due to distances. I would like to say one more time, thank you for the Dr. Wang, because he kind of shifted gear from a bit of a classic looking, where I would go for and talk the similar way. Regarding telomeres, you know, it's not a new topic actually, and many of you know in advance, and it has been quite well known, and there has been in the field basically 4 or 5 Nobel Prize winner in that sense. So it is why it is important, why does it matter, and why are we still talking about telomeres? You could start to question it, because we all know about that. It is like the telomeres has been pronounced and has been followed that they get shortened with every cell division, with the damage too, and they have implication on the associated aging related diseases. And of course, if you couldn't prevent that, and if it keeps continuing as it's been calculated, you expect that it comes to a level where it shortens so much that it reaches the coding genetic sequences where it actually can be quite fatal or really severe consequences for the organisms. So therefore, there is a way, and the mechanism has been developed that telomeres are inducing cell replication to stop. And if they cannot do that, and it leads to a different kind of possibilities, different diseases like a cancer, because they have an escape pathway. But then of course, what do we know of until now? And there has been so far identified many diseases that, especially it was the group of the premature aging disease group, and slowly within the last decade, we turn back again, start to highlight the telomere issue, where we start to use, and even companies built and established based on that, looking for the can telomere length allow us to in some way distribute or tell about our future. Can you use the prediction about your future disease and your remaining lifespan? And when you look at it, the new disease group is looking at the degenerative joint diseases, sensory impairment diseases, cancer has been there always, and inflammatory disorders. So we thought, why is that? We is still just like Dr. Wang's presentation, we have the old information, but then we can see something is not really matched always. And what we knew in advance, and we look for the overall telomere length. We knew that the telomeres get shortened or decrease with age, and has been calculated for a different cell types. For example, it could differ from the approximately 60 base pairs per year to could be a 40 or sometimes less, sometimes more. But then the surprise part that even the non-dividing cells are shortened. So that for me, it kind of bring a hint, okay, are we really looking at the right way of evaluating the telomeres? How come if it is only related to dividing cells shortened, how come the non-dividing cells shortened? So maybe we are not evaluating them right. And we look at what is out there. Currently, there are several methods that you could use for evaluating telomeres. One of them, the very first one, was the Q-FISH, where it looked for the total or a single telomere length in a single cell. You get the mean average of the telomere cells, and but the disadvantage that it required always a viable cells. And then there was a qPCR based telomere length measurements. It was much faster compared to that one, but then there was a variation between the groups. Then this is the most often used telomere restriction fragment method, and it also gives you the mean telomere length. It's so far it has been accepted as a gold standard, and it only detects the mean for the given sample. So we said, okay, that gives some important information, but we need to do something bit more on top of it. So we actually start to use our ideas, trying to use this quantitative fish on a digital image analysis system. And then when you look carefully, you could see even on the same chromosome, one was highly expressed, you could see the telomeres, and the other one basically was like the signal is not there, or it's been really shortened. So we tried to do some calculations again, and then to get a 20 cells from a 20 different individuals to make a mean telomere evaluation to get it sorted out and standardized. So if everything was fine, you could see this profile mathematically. You could calculate on this imaging system that there is a difference, and it perfectly matches for example, the tall chromosomes has the tallest telomeres, and the short ones has the short ones, and the long arms, short arms. You could see there was a profile. And then when we tried to do it on a 20 individuals and we get the normalized according to, we could create a basically 95% with this sort of the intervals for that. And if everything was supposed to as it goes, like according to this calculation, you would have a telomere profile, if you sort it out according to length, is a linear line. And this was what has been always discussed. But we were first time could say, okay, we could show it. Then we turn and take samples for the aging groups. Oops, sorry. Aging groups, and you could, in reality, what you see in this first bar histogram is there, that you could see then if you take the young people and the relatively what you say the elderly people and then very old people, there is you could see this telomere length changes or goes like scale to the right with age. But then if you put the profiles, you could easily question me, but it doesn't look like they're fitting. It's because you had to normalize them for this average. Once you normalize, you could see that yes, it does follow the normal telomere profile, and still you could see the shortening. So it allows us to say, okay, we're in the right direction and we can detect it. So we wanted to further develop into more precise and more specific. Then we used PCR principle on a chromosome analysis. So we developed something called dideoxyprimed in situ labeling, which is basically you do a one cyclus PCR with a both mix of the labeled and unlabeled nucleotides. And then of course, between the competition among these labeled and non-labeled, you end up seeing much more precise compared to FISH approach, and even standard prints. You could see the dideoxy gets much more specifically, and those that were really short and in reality you couldn't detect them, you are capable of detecting in these approach. So this was the first on the FISH. When you mark it, you couldn't see it, but then if you apply the dideoxy prints, you can highlight and see these small ones. But of course, this is a very labor demanding and time consuming to do it every single 92 end of the chromosomes and for many samples. So to do it even larger scale, we decide to further develop this idea by developing an approach called universal stella. It's basically we got the print. It's the same principle as has been Carlton has identified X and Y stella, and that was based on only 2 chromosomes, and we have done it for the 92 end. The difference here was the principle here. You could use a G-rich 3' overhang, and then allows you to use this kind of a linker telluride, and as after you isolate the sample DNA, and it allows you to use this as after the digestion you have this telluride as a primer, and then you could use an adapter for it and amplify it by several PCR cycles. And by the end of it, this ensures that you get all the very short ends that you were never capable of seeing, you could see them. To identify that this principle worked, and we were identifying the really short ones that we were not seeing in reality with the other techniques, we used the mesenchymal stem cells, where we had the different lineages of they were first, you got the primaries, and also we tried based on a telomerized, which they could continue to population doubling level up to 45. Normally primaries wouldn't continue more than 10 doubling levels, so you could increase it. But that allows you, by taking different population doubling levels and applying this universal stella, you could see them normally what we'd see in the pickup in the TRF assay and every other assay, all the telomeres length is there. But all of a sudden, what we couldn't detect, and perhaps that's the reason that all these aging-related disorders, including cancer, that we couldn't detect until it's too late with the telomere length, there's lots of ultra-short telomeres, and that gradually with the increasing with the population doubling levels. So you can see it's at the very beginning, you have very few, almost none. Then the further up you go, you could see the more and more ultra-short telomeres comes up. To correspond, this was fitting with the aging of these particular stem cells. We combine it and overlap to each other with the beta-gal stainings and associated beta-galactosidase staining, and you could see this profile fit perfectly. The old the cells get, the more senescence the cells get, the more ultra short telomeres you could detect. So we said, how about this was done on a cellular system? How about detecting this in the actual diseases like articular cartilage, which is normally based on stress and overweight, and generally people develop that with the age. We took these knee replacement therapy operations, and then you take these samples divided into four, based on the damage or a stress induced parts that you got it from 1 to 4, and isolate the DNAs from there, and try to, based on the zonal distribution, look for the ultra-short, the presence of the ultra-short telomeres. And then, of course, the number 4, where you had the highest damage, you actually see again the accumulation of the ultra-short telomeres, which also showed that yes, maybe, but same people, if you take the blood samples, you wouldn't see as much telomere shortening where you take only the mean value of it, another organ. So if you look at it, actually, that it tells you something about it. It's always significant to evaluate the relevant organ. Then we say, okay, we take another sample. Here you could discuss that there was also physical damage. We take the COPD patients, which is also known that often you develop it, heavy smokings and the low lung functions and age. So it is another age-related disorder, and it's one of the main reasons that you lose people apart, besides cancer. So here we design in a way that we collect the samples. Those people who were having cough, constantly coughing for 3 weeks with no explanation, and there has been a decision by the clinicians that they will go for a bronchoscopy. So during that procedure, we asked them to give us a bronchial lavage samples from the same people. Same day, we also collected some blood samples to evaluate the peripheral blood telomere lengths. And we look at in these 2 different samples from the same people, together with their lung function and so on. This was the clinical part has been done to say, yes, these people had the COPD. So our results actually showed that presence of the ultra-short telomeres in leukocytes, the same persons, was much less compared to if you look at the bronchial lavage samples. So that again emphasizes and tells that looking at the ultra-short telomeres and their presence is much more informative. And if you do it on the damaged tissue or the targeted tissue, you get more information. So it's not about only looking overall shortening, it's looking at their profile, how they develop. So of course, this study was different from the literature in a 2 ways. One of them, until now, we always talk about the mean telomere length. So here we are talking about the presence of the ultra-short telomeres. And again, until now, we always look at the leukocytes, like the peripheral blood, but this time we are emphasizing that, well, diseases like COPD, which there is a high inflammation, age-related disease, if you look at the related organ or a tissue, you can get much more information, and you can actually see that every single telomere end is shorter than approximately 1500 base pair. You could detect and direct associated. Of course, to try to make it more correct, we also try to take the samples, biopsy samples, when it was taken, or bronchial lavages, put them on a lung map, kind of, and see if it was have anything to do where it's been taken. So it means that if it was organ specific, does it also the part of the organ or the tissue, does it have anything to do with it? So we could say that there was no bias for that. And it was overall, you get the information when you have the valve anywhere of the lung, you could get the right information and the right shortening. So this actually told us that overall, we have the information about it. Looking at the tissue-specific sampling is extremely important if you're dealing with the senescence-associated aging-related disease like cancer also. And looking at the tissue-specific ultra-short telomeres, you can use them as a potential biomarkers, where so far mean telomere length has been associated with the disease, but you could not really use it as a potential biomarkers, but it's still, this time, if you're using the ultra short ones, you can use them as a potential biomarkers. And then it can also tell you something about that the disease's severity and the stage, because you have the chance of calculating or counting how many ultra short telomeres you have. So we took the same information in the cancer perspective. Again, take the mesenchymal stem cells, and then even though what we know about it, stem cells, they are not always, unless they are required, they don't not divide as well. So we could kind of say that they were this non-dividing cell group. And of course, they also show senescence. We could see if you are having in the culture, but if you look at it from the, for example, knee example, you could see that there is also getting aging procedure. And another proof was, especially for the, when they are talking about mesenchymal stem cells, you have the, if you look at the bone, you can see that with the age, the bone mass is sort of getting less and less compact, which means that there is a proving that there is a aging and then cell losing there. So we took these particular mesenchymal stem cells, we took 30 young donor and 30 old donor, and look at their telomere profiles. And again, try to compare this with the senescence association with the telomere profiles. And you could see the, yes, the old ones had the more senescence, and then the shorter, ultra short telomeres, and the young group had the much less senescence and better telomere profile. So once we had these samples, we say, okay, on particular in these samples, we want to study the neoplastic transformation, like in the cancer development within the mesenchymal stem cells. So we took these cells and radiate them to different gray. One was 2.5 gray and the other group was 15 gray. And when you look at it, in reality, all these had a normal, we're pretty much matching the leukocytes, the first profile that we identified. They had the normal expected telomere profile, but after we exposed them to irradiation, the profile has changed. So there has been a damage. As Elizabeth Blackburn underlined, once you are stressing the cells, you can have abrupt shortening. So it's faster shortening. And you could have a good explanation or a mechanism to study if it was this abrupt shortening and telomere shortening leading for a tumor formation. So we took those samples after we irradiate them with both to 2.5 gray and 15, but then 15 gray where we could see that in the culture they were not particularly, they got so much damage that they couldn't really divide. So we continue with the in vivo tumorigenicity assays by taking the subclone of irradiated cells, putting under the skid mice, under their skin, and observe it for 6 months. All of a sudden, after 6 months, as a long-term effect, those cells who had lost the profile, not as much as the 15 gray, but they had a certain amount of damage, we could see that there was a tumor development on a back of a skid mice. And then we took those and tried to compare it for the, again, controlled primary MSCs and the non-irradiated MSCs, and then the different gray irradiated MSCs in different perspective. And then we could see that, yes, slowly it led to accelerated telomere shortening. So there was some damage, and this was the explanation, or you could say this was the initiating explanation for that. What we know about these, normal what you do, you look for also the, after the irradiation, you look for the increased level of NFS breaches, which is a well-known thing. And slowly we could see, in the, if you do the chromosomes, you could see from metaphases, slowly the balance shifts towards the unbalance, because there was slowly translocation, big translocations. Then it was much more small breakages, whole part of the chromosome or a partial chromosomes, or the whole arm of the chromosomes, and then slowly it goes like the losses and the gains. So you could see that genomic shifting imbalance was appearing. So putting all these together into a concept, what we learn, so you know that normally mesenchymal stem cells and every other stem cells, they have this high self-renewal and they should repair themselves, right? But once you are having this accelerated telomere shortening, or you create a damage on that, it will be resembling like during the aging period. It's like normally the cells should go through the P53, RB, or a P16 checkpoints, and if they can get repaired, it continues gradually aging. If not, they should go for a normal senescence or stop dividing. So instead of stop dividing, in this case, what happens, they adapted it, these chromosome ends broken, then they create a chromosome fusion, and the fusion break fusion. Because how do we know that? We know it through the NFS breaches. And every time there was a new break, they change, had a bit more telomere or chromosome piece for transfer to the other chromosome. And then it was like leading slowly a more imbalanced genome. So what we call it, the chromosome breakages, fusion breach cycle came on. And after this cycle, of course, these cells learn to survive, but they become cancerogous. So this gave them a more resistance, and they are more strong against to most of the treatment approaches also. And this way you ended up seeing the telomeres in directly relation to cancer. So, which tells us that as we started initially, you can have many different methods to, but you have to choose your method based on the question you wanted to address. And if you wanted to actually talk about the aging related diseases like either cancer or inflammatory diseases or arthritis or COPD, it's a good idea to use a universal stela where you could actually really evaluate the ultra short telomeres, and you can get information about the current state of the disease, diagnostically and prognostically. And you could talk about that, yes, there is an initiation or the ongoing state of the disease. So basically with this presentation, what my message is to you that, yes, telomeres have important functions all throughout our lives, and they are influenced or causing a different cellular problems. But looking at the white blood cells, like leukocytes, it only gives you a proximity and should be more specifically evaluated. So as a person who is working with the telomeres and stem cells, even so I say, yes, looking at the white blood cells, sometimes we have actually said that we do not see any association within the telomere length, is probably because we evaluate the wrong samples. If you're looking the right samples, you can use the telomeres as a good clinical parameter. And instead of looking at the overall telomere length, we should look at the number of the short telomeres, ultra short telomeres. And there you could get a much more information based on the defective hormone and more straightforward information for that. So that's all what I would like to say about it, and I'm willing to take your questions. Thank you very much for your patience.